Okay, so we've spent the last section really focusing on DNA, what it is, what it looks like. So now we're introducing yet another nucleic acid, and now we're talking about RNA instead. So RNA stands for ribonucleic acid, and its function really is to take that information that's coded for, housed in your DNA, and use that information now to help build these proteins. So if we look, this is an RNA nucleotide. It looks very similar to what we talked about with the nucleotide structure of DNA. I've got my phosphate group here attached to my 5' carbon. I've got my sugar. It's still got 5 carbons, and I've got my nitrogenous bases bound to that first prime carbon. Couple of differences that hopefully you notice. All right, in this case, my sugar is ribose as opposed to deoxyribose. If I wanted to dig into this structure a little further, what I would see here in this ribose is that there's an OH off the second prime carbon as opposed to the deoxyribose that's missing that oxygen. So deoxyribose only has a hydrogen here, hence the deoxygenated part of that name. Also, if I look at my nitrogenous bases, I still have an adenine, a guanine, and a cytosine, but you notice that thymine is missing. And instead of thymine, we've got a U, which stands for uracil. So here are your big picture comparisons. So the sugars are different. DNA is the deoxyribose, and RNA it's ribose. The other thing is how many strands they have. So DNA is double-stranded, RNA is single-stranded, at least in most situations, in most cells. And then you've got your bases, right? So the A, C, and G are still there, but now I've got thymine strictly in my DNA, uracil strictly in my RNA. And there are 3 unique types of RNA based on what they do in your cells. So when we talk about that idea of flow of information from DNA to RNA to protein, that particular RNA molecule that we're usually referring to, that copy of DNA information, really is messenger RNA, or we shorthand it to mRNA. So when we talk about going through transcription, making that copy of that DNA gene, that DNA code, we are creating messenger RNA as a result. Now, here's the interesting thing. When we look at DNA and RNA as a result of that, everything is in code, literally 3 bases at a time. So we read it as a triplet code. And every 3 bases in DNA, and as a result, every 3 corresponding bases in RNA, is what we call a codon. And think about it if you've ever done a cipher puzzle before, where you had numbers or letters that corresponded to something else and ultimately gave you a message at the end. So for a codon, every 3 bases in either my DNA or my RNA will ultimately give me the information for a single amino acid. So that means a 12 base pair code gives me the information to make 4 amino acids. When we get to the nitty-gritty details of how translation works in a little while, what we'll talk about is this codon chart and how to read it. When we look at it, there are 20 different amino acids, which means there has to be at least 20 different codons. 20 different triplet codes that give me the information for those amino acids. There are actually more than 20 codons, partly because there's some redundancy or duplicates, if you will, in the code, and we'll see that when we get to that point. But there are also codons, there are also base codes that tell us where to start and where to stop. So when we think about your code, think about it almost like a really simplistic sentence. So I've got a capital letter so that I know where it starts. Well, there's a start codon, so I know where the information starts. There's a punctuation at the end, hence the stop codon, so I know where everything stops. And then you've got all the good information that gives you the message of that sentence in between, and that would be what would actually translate into amino acids, and as a result, build the protein that we're trying to create. Second type of RNA is transfer RNA or tRNA. These guys, cleverly enough, not only do they transfer amino acids around the cytoplasm, but they actually look like T's, which is kind of crazy and kind of cool. That particular RNA molecule, this is a single strand of RNA, but it bends back on itself and binds to itself, creating this T structure. Now, when we look at this transfer RNA, its job is literally to transfer, to move amino acids around the cytoplasm. So these tRNAs are floating around your cells. They're picking up amino acids that your body has digested and broken down from things like the food that you've eaten, attach them to themselves, and then we'll take them to the ribosomes to actually help build the protein itself. Now, how does that tRNA know which amino acid to collect? Well, they've got a binding site here for that amino acid, and they also have a binding site down here. This is something called that anticodon, and this is 3 bases of RNA that complementarily base pair to the codon that we see in your messenger RNA that also complementarily codes to the codon in DNA. So if I have an anticodon here, that would be CCG. That means the codon that it creates or that it connects to, if this is CGG, then I've got GCC. Okay, so they're opposites, they're base pairs. So you got to remember your base pairing rules. Only difference here is that A still goes with C, but now in RNA, A goes to U. And we'll do some practice with that in the coming, in a little while. So remember that amino acids are your building blocks for proteins. So the whole idea here is to pull those amino acids together, build the proteins that your cells need to function, whether those are structural proteins, whether they're enzymes, transport proteins, whatever the case might be. Your cells all work off of these functional proteins, so we need to create them. So, just like I mentioned a few moments ago, that there needs to be a particular codon, a 3-base code associated with every amino acid, that also means there has to be at least a single tRNA for every amino acid as well. And just like there's multiple codons for the same amino acid, that also means there are multiple tRNAs for the same amino acid, but there has to be at least 20 to make this all work. Third type of RNA is ribosomal RNA. This one's really just more structural. We shorthand it to rRNA, and this ribosomal RNA combines with proteins in the nucleolus in the center of your nucleus. When we go to actually form those ribosomes, your ribosomes, we're going to see a little later, actually come in 2 subunits, so a big subunit and a small one. They kind of remind me, at least in the diagrams, of a burger roll, kind of big and fluffy at top and flat on the bottom. But regardless, your RNA, ribosomal RNA, really is just a structural component to building those pieces along with some other proteins.